Imagine for a second, right, that you've been hired as the lead architect to build this massive skyscraper.
Right, that skyscraper.
Yeah. But there is a catch.
Sure.
So instead of just, finishing the roof and calling it a day, your goal is to keep adding floors to this building forever.
Oh, wow.
Right. You are building an infinite tower. And while your central construction crew is just endlessly building upward into the clouds, you also have to build finished, completed balconies on the sides of every single new floor.
I mean, that sounds like an absolute logistical nightmare.
Well, it gets worse. Because here's the real puzzle. How does the original architectural blueprint actually know which part of the construction crew needs to stop, put down their tools, and build a terminal balcony, and which part needs to just keep laying bricks upward indefinitely?
Because if those instructions get crossed, right?
Yeah.
If the upward building crew gets confused and builds a finished balcony right on top of the main tower, the whole project terminates. The skyscraper just stops growing completely. It's a massive coordination problem.
And today, we're taking a deep dive into how nature solves this exact architectural puzzle. We're looking at a truly fascinating paper published in the journal Science by Tian Huang, Doris Wagner, and their colleagues.
All right, yeah.
Now, the original title of the paper is a bit of an academic mouthful. It's called A Negative Feedback Loop Between Terminal Flower1 and LEAFY Protects Inflorescence Indeterminacy.
Which is a lot of words.
It is a lot of words. But at its core, this research basically decodes nature's ultimate architectural algorithm.
That is, I think that's the perfect way to frame it. What this team of researchers managed to do is uncover the microscopic blueprints of exactly how a plant grows.
Yeah, so our mission today is to unpack every conceivable detail of how a plant decides to keep growing its main stem, which biologists call indeterminacy,
Right.
while simultaneously turning its side shoots into flowers, which is determinacy. And specifically, we want to understand how it handles incredibly noisy, chaotic environmental signals without, messing up this delicate architecture.
It's a huge task.
It is, but we have the perfect expert guide here to help us navigate these complex biological algorithms.
Well, thank you. I mean, to really grasp what is happening here, we kind of have to start by looking at a fundamental biological paradox.
Okay, lay it on me.
Picture the very tip of a growing plant shoot. Up there, you have two neighboring groups of stem cells. They are practically touching one another.
Right.
And in the spring, when the days get longer, they receive the exact same seasonal signal. But here's the paradox. Despite getting the exact same signal at the exact same time, they make completely opposite life choices.
Really? The exact same signal?
Exact same. One group decides to become a flower, which essentially means it stops growing and eventually dies. And the other group decides to remain as stem cells and just live on forever.
Okay, let's unpack this.
Yeah.
Because to really visualize this paradox, we need to know the players on the board.
Yeah, definitely.
Let's start with the architecture itself. When you say stem cells, what are we actually looking at?
Right, so in plant biology, a pool of stem cells is called a meristem. Think of it as a reservoir of raw, unassigned potential.
Got it.
At the very top of the growing shoot, you have the inflorescence meristem, or IM.
Okay, I am.
The IM is your upward building construction crew. Its entire job is continuous, infinite growth. It just keeps pushing the plant taller and taller. That is what we mean by indeterminate.
Okay, and then on the flanks, right, on the sides of that main upward growing stem, we have the lateral meristems, the LM.
Yes, exactly. The lateral meristems are the balconies in your skyscraper analogy.
Nice.
They are determinate. Their destiny is to stop growing, build a highly complex structure, a flower, and ultimately produce seeds. So that is our physical spatial setup.
Right.
But to understand how they know what to do, we have to look at the chemical signals moving through the plant. The massive systemic trigger for all of this is a protein called FT.
FT?
Yeah, which stands for flowering locust T. It's often just called florigen.
Florigen, I love that name. It sounds like a mythical potion or something. The flower maker.
It basically is. When the environment changes, say, the days get longer after a dark winter, which provides a far red enriched photoperiod, the leaves start pumping out this FT protein.
Okay.
FT then embarks on this epic journey all the way up the stem to the very top to deliver a systemic message to those stem cells. And that message is, winter is over, the time to make flowers is right now.
So FT is the messenger, and when it arrives at the top, it wakes up our next major player, right? A pioneer transcription factor called LFY, or LEAFY.
Exactly.
I think of LFY as the foreman of the balcony crew. This protein desperately wants to turn stem cells into flowers. It exists to promote floral fate.
But here's where we run into a major issue. If LFY had its way completely, the plant would just turn into one giant explosive flower and stop growing entirely.
Oh, wow.
Yeah, that upward building engine would just crash, so there has to be a counterbalance. And as our final core player, TFL1, or terminal flower1.
Terminal flower1, okay.
TFL1 is a core pressor. It's the ultimate protector of the stem cells. Its sole job is to promote indeterminacy and strictly stop flowers from forming in that main inflorescence meristem.
I like to think of this actually as a train laying its own tracks into the wilderness.
I like that.
Yeah, so the inflorescence meristem, the IM, is the engine at the front, laying tracks endlessly into the future. The lateral meristems are the little train stations being built along the sides of the tracks.
Right.
But wait, if I can just push back on this whole setup for a second.
Sure.
If that seasonal FT signal floods the entire system, and FT tells absolutely everything to make flowers by activating LFY, why doesn't the main engine just stop, build a station right on the tracks, and turn into a giant terminal flower?
Yeah.
Like, why doesn't the whole plant just commit to flowering?
I mean, that is the multi-million-dollar question in plant biology. And to figure out how the system survives that massive FT signal, the researchers looked at what happens when the system physically breaks.
Oh, by looking at mutants.
Exactly. They examined mutant plants. If a plant has a genetic mutation where it loses that protector protein, TFL1, this is called the TFL1 mutant, the plant does exactly what you just suggested.
Wait, really?
Yeah. The main inflorescence meristem terminates immediately. It literally sprouts a single flower at the very top and completely stops growing. The engine derails.
Wow.
Conversely, if a plant loses LFY, the LFI1 mutant, it just endlessly grows upward, but struggles to ever make proper flowers on the sides.
It is such a delicate, high-stakes balance. So let me just do a quick recap here to make sure I'm holding all these pieces together and for you listening as well.
Good idea.
Basically, bullet point 1,
Yep.
we have the main stem, the IM, which equals continuous growth or indeterminacy.
Yep.
Bullet point 2, we have the side stems, the LM, which become flowers, so determinacy.
Right.
And bullet point 3, FT triggers LFY, which is the flower maker, while TFL1 is the stem cell protector.
That is the perfect mental map. And honestly, it makes you wonder about human nature, too.
How so?
Well, think about your own life for a moment. When you receive a massive external signal or pressure to change everything about your environment, what is your internal TFL1? Like, what is that core identity protector that keeps you moving forward on your main track?
Oh, man. I love that, holding on to the core engine while still building the lateral balconies.
Exactly.
Now, here is where it gets really interesting, because the researchers didn't just stop at identifying these proteins, right? They wanted to see the actual wiring.
They did.
They zoomed all the way down to the cis-regulatory regions, the genetic control switches of the TFL1 gene itself, and they found a massive plot twist.
They really did. So they mapped the genome and focused on one incredibly specific 527 base pair sequence called region 7. Yeah, it sits right in the regulatory area of the TFL1 gene. You can think of it like the main breaker switch in an electrical panel.
Okay, making sense.
To test what it did, they used CRISPR gene editing, which acts like highly precise molecular scissors, to snip out just this specific region 7, creating a new mutant called CRIFN.
And what happened to the plant when they snipped that specific wire?
Total systemic failure. The plant immediately grew a terminal flower at the top. The main inflorescence meristem just died.
Okay, so region 7 is the absolute crucial control switch. It has to be flipped on to protect the stem cells.
Exactly.
But here's the paradox. To figure out what was naturally flipping that switch in a healthy plant, the team used a technique to see exactly which proteins are physically stuck to the DNA at any given moment.
Yes, they used ChIP-seq. It's a brilliant molecular tool. It allows scientists to stop guessing and actually see, like, the physical interaction between a protein and a DNA sequence, a sort of molecular freeze frame.
And when they ran this analysis, they found out what was physically binding to region 7. It was LFY.
Which is wild.
The very protein that wants to make flowers is physically binding to the DNA to turn on TFL1, the protein that stops flowers.
It seems completely backwards, doesn't it?
Totally.
The researchers knew this was incredibly counterintuitive. So to prove they weren't just seeing, experimental noise, they used some highly advanced visualization techniques.
Right, because seeing is believing.
Exactly. Instead of just grinding up the plant in a blender to see what chemicals were inside, they basically turned the inside of the cell into a glowing neon sign.
The imaging they achieved here is mind-blowing. They used HCR RNA, FESA-H, which is a highly sensitive fluorescence technique, right?
Yes.
And they combined it with IHC, immunohistochemistry. They made the specific messenger RNA molecules glow one color, and the actual proteins glow a different color.
Right. So they were looking at intact, single optical sections of the microscopic plant under a confocal laser microscope. They literally caught them in the act.
And what exactly did they see?
When they look at the very center of the inflorescence meristem, they saw the LFY protein and the TFL1 messenger RNA perfectly overlapping. The glowing colors literally merged. They were occupying the exact same space at the exact same time. The flower promoter was directly standing on the switch to activate the flower blocker.
I was trying to wrap my head around this, and here's a memory trigger that helped me.
Oh, let's hear it.
Think of LFY as a hyperactive party host. He wants to throw the biggest, most chaotic floral party ever, but he knows his own tendencies. He knows that if he gets too excited, the party gets completely out of hand, the authorities are called, and the whole venue is shut down permanently.
Meaning the plant makes a terminal flower and dies.
Exactly. So the very first thing this hyperactive host does when he enters the main VIP room, the inflorescence meristem, is hire a massive bouncer. And that bouncer is TFL1.
I love this.
Right. LFY hires TFL1 to keep himself in check.
That analogy actually maps perfectly to the molecular mechanism. The party host has to hire his own restriction to ensure the venue survives the night.
So if we're doing another quick bullet summary of this incredible twist. Bullet 1. Region 7th is the master switch for TFL1.
Yep.
Bullet 2. LFY physically walks over and binds to region the 7th.
Right. Bullet 3, the paradox. The flower promoter activates its own worst enemy, which leaves you with another fascinating engineering question to ponder, right? If you were designing a complex system from scratch, why on earth would you ever wire the on switch directly to the off switch?
Seriously. It sounds like a terrible way to wire a house. If I turn on the lights and it instantly triggers the breaker to shut the house down, I'm firing the electrician.
Yeah, you'd be pretty mad.
So what does this all mean? How does this bizarre wiring actually benefit the plant?
Well, what looks like a flaw is actually brilliant engineering. In biology, this is called a negative feedback loop.
Right.
It is a highly sophisticated algorithm designed entirely for robustness. It buffers the system against chaotic noise. And to truly understand it, the research team realized they couldn't just look at plants in a greenhouse anymore.
What did they do?
They had to build complex mathematical computer models.
Math to the rescue. Biological algorithms.
Precisely. They used specialized delay differential equations to simulate thousands of possible parameters.
Wow, thousands.
Yeah. They started with what they called a two-node model. Node 1 is LFY. Node 2 is TFL1. LFY turns on TFL1, and TFL1, in turn, represses LFY. It is a closed infinite loop.
Okay, that makes sense.
Then they upgraded the math to a three-node model by adding FT to the mix.
Right, FT is the chaotic seasonal signal coming from the environment.
Exactly. Imagine a sudden massive spike in FT because the environment is suddenly absolutely perfect for flowering. Maybe there's an intense 24-hour far-red photoperiod pulse.
A huge blast of spring.
Right. FT floods into the stem cells and aggressively turns on LFY. Because of this, LFY levels spike dangerously high. The party's about to get out of control.
Okay, I'm with you.
But because of this negative feedback loop, that massive spike in LFY immediately turns on a proportional massive spike in TFL1. TFL1 then acts as the bouncer, grabbing LFY and violently pushing its levels back down to a safe, steady state.
Okay, I follow the math in a computer simulation. But how do you prove a theoretical math model inside a living, breathing plant? I mean, plants grow slowly. You can't just wait around to see if the math is right.
You are absolutely right, which is why they had to use real-time chemical induction. They essentially engineered genetic tripwires into the plants.
Wait, genetic tripwires?
Yeah. This allowed them to artificially break the nodes one by one on command. First, they engineered a plant with a dexamethasone-inducible mRNA.
Okay, that sounds like molecular warfare. What does an inducible mRNA actually do?
It acts like a targeted assassin. They spray the plant with a specific chemical trigger, dexamethasone. When that chemical hits the cells, it activates the artificial microRNA, which instantly seeks out and destroys the LFY transcripts.
Oh, wow. And the result?
The moment LFY was assassinated, TFL1 levels plummeted. Without the party host there to hire him, the bouncer immediately goes home. This proved the first half of the math. LFY absolutely drives TFL1.
That is wild. And then they did the exact reverse, right?
Yes, exactly. They engineered a different tripwire, an estradiol-inducible GFP degron.
Another mouthful.
Right, but same concept. This time, they sprayed a different chemical, estradiol, which instantly hunted down and destroyed the TFL1 protein.
Okay.
And the result of that, LFY levels absolutely skyrocketed off the charts. Without the bouncer there to keep order, the party host goes completely wild.
So they proved it.
Yes, this proved the second half of the math. TFL1 is actively and continuously repressing LFY.
So what does this all mean for the plant? It's exactly like noise-canceling headphones.
Oh, that's a great way to put it.
Right. Think about it. When you put on a pair of high-end noise-canceling headphones, there is a tiny microphone on the outside listening to the chaotic noise of the airplane cabin or the street.
Yeah.
That external noise is the FT seasonal signal. The louder that outside noise gets, the stronger the headphones generate an exact inverse sound wave.
And that inverse wave is TFL1.
Exactly. The inverse wave completely cancels out the noise to protect your ear. And in this case, the ear is the delicate pool of stem cells at the top of the plant.
What I love about that analogy is that it highlights a crucial point. The system isn't just ignoring the environment.
Right.
It is actively listening to it. It is using the energy of the noise to generate a proportional countersignal to maintain internal silence and stability.
So if I'm summarizing this mechanism for you listening, bullet 1, LFY and TFL1 are locked in this highly controlled, closed, negative feedback loop.
Yep.
Bullet 2, computer simulations with thousands of parameter sets accurately predicted how these proteins behave under stress.
Right.
And bullet 3, real-life chemical induction proof the math. Destroying the microphone stops the inverse wave, and destroying the inverse wave blows out the speaker.
It is the ultimate biological shock absorber. And when you think about it, it makes you wonder where else in nature or even in our own bodies do we have these invisible negative feedback loops?
Yeah, that's a profound thought.
Whether it's regulating your blood sugar after a massive meal or maintaining your core body temperature in a blizzard, these loops are the invisible bouncers keeping our systems from crashing every single day.
It really is the hidden secret to biological stability, but I have a major pushback here.
Okay, let's hear it.
Something in this blueprint isn't adding up for me. If the FT signal is moving everywhere throughout the tip of the shoot and LFY is getting turned on everywhere by that signal,
Yeah.
why does this noise-canceling loop only happen in the main inflorescence meristem? Why don't the lateral meristems, the balconies on the sides, also turn on TFL1, cancel out the noise, and just keep growing into endless upward branches instead of becoming flowers?
That right there is the spatial paradox.
It's a glaring hole.
It is.
Yep.
And it was the final, most complex piece of the puzzle the researchers had to solve. They realized their 3-node model was beautifully accurate for the main stem, but it wasn't complete enough to explain the whole plant.
Okay, so what did they do?
So they expanded to a 4-node model. They realized there must be a mechanism to physically break the loop in specific locations. There had to be an exit strategy.
An exit strategy, because, if there isn't, the plant never makes a flower, never reproduces, and the species just goes extinct.
Exactly. The mathematical model predicted that if LFY levels manage to get astronomically high, the loop breaks. TFL1 doesn't just fail to keep up, its levels actually drop.
Really?
Yeah. The math demanded that there must be a hypothetical factor X at play. Factor X is an unknown repressor protein.
And this invisible factor X is hiding out in the lateral meristems.
That is exactly what the model suggests. In the specific spatial geographic context of the lateral meristems, this factor X is present in high amounts.
Okay, walk me through it.
So let's play out the scenario. The seasonal FT signal hits the lateral sides. LFY wakes up and gets excited.
The party host is ready.
Right. But before LFY can hire its bouncer, before it can walk over, bind to region 7, and flip the switch to activate TFL1 factor X, steps in and intercepts it. It physically blocks LFY from turning on the protection system.
Oh, wow. So in the side balconies, LFY is completely prevented from turning on the noise-canceling headphones.
Correct. And without TFL1 there to repress it, LFY runs completely rampant. It hits that astronomical saturation point, and the stem cells in the lateral meristem rapidly and irreversibly commit to their fate.
They become a flower.
Determinacy is achieved.
What is so fascinating here is how incredibly elegant that solution is. It completely explains two deeply contradictory concepts in biology, right? Phenotypic plasticity and robustness.
Yes. If we connect this to the bigger picture of evolution, this is the holy grail of survival.
How so?
Well, phenotypic plasticity is the ability to flexibly adapt to the environment. The plant senses the long days of spring and exhibits extreme flexibility by quickly building flowers on its lateral side so it can reproduce.
Right.
But at the exact same time, it requires robustness, an ironclad, mathematically perfect protection of its future growth potential in the main stem, regardless of how loud or chaotic the environmental signals get.
It essentially dual wires the exact same chemical signal to mean two completely opposite things, based entirely on the geographic location of the cell within the plant.
Exactly.
So wrapping up this final piece for you, bullet 1. The noise-canceling feedback loop is strictly context-specific. It's only allowed to function in the main stem.
Yep.
Bullet 2. A hypothetical factor X in the side stems physically intercepts the loop, allowing those cells to achieve their floral destiny.
Right.
And bullet 3, this dual wiring is what allows the plant to be incredibly robust and flexibly reproductive all at once.
Which brings up a really compelling thought experiment for you. Imagine if human cells had their own versions of a factor X.
Oh, man.
Right? A molecular mechanism that could tell identical stem cells to build a complex organ in one specific geographic spot in the body, but strictly stop them from growing endlessly in another.
That would change everything.
How might uncovering these localized, context-specific loops fundamentally change the future of regenerative medicine, or even how we approach cancer?
That is profound, truly. So, to synthesize everything we've explored in this deep dive, we started out as architects looking at the macroscopic structure of a plant, just trying to build a skyscraper with infinite balconies.
Yeah, a seemingly impossible task.
And then we zoomed all the way down into the genome to a microscopic 527 base pair genetic switch.
Region 7.
Right. We uncovered an elegant mathematical negative feedback loop. We verified it with glowing neon RNA under a laser microscope, and we discovered how a plant uses spatial geography and a mysterious interceptor called factor X to decode a single, chaotic environmental signal into two completely different biological destinies.
It truly is a masterclass in biological data processing.
Yeah, it is.
The plant is essentially a supercomputer making life and death decisions based on fractional changes in protein levels. And it leaves us with a final, somewhat provocative thought to mull over.
Let's hear it.
If simple plants evolved this highly sophisticated, mathematically perfect negative feedback loop to protect their stem cells from environmental noise, it raises an incredibly important question. Do our own human stem cells have similar undiscovered noise-canceling genetic loops? Invisible bouncers that prevent us from aging too quickly or mutating when we are exposed to toxic environmental stress.
Wow.
And if we could fully map them the way these researchers did with Regions the 7th, could we rewrite our own biological blueprints?
Could we learn to keep laying our own tracks forever?
Exactly.
That is something to think about. Thank you so much for joining us on this deep dive. As always, keep questioning the blueprints of the world around you, and remember, sometimes the best way to handle the noise of the world isn't to hide from it, but to build a better pair of headphones. We'll catch you next time.